Good day, sir. By the way, I am Junel Fortune Megallen Jr. And the topic that we are going to discuss to you, sir, it's all about what are the risks and challenges of gene therapy and stem cell research. Gene therapy involves altering the genes inside your body cells in an effort to treat or stop diseases. Gene therapy has some potential risk. A gene cannot easily be inserted directly into your body cells. Rather, it usually has to be delivered using a carrier called a vector. The most common gene therapy vectors are viruses because they recognize certain cells and carry genetic materials into the cell's genes. Researchers remove the original diseases-causing genes from the viruses, replacing them with the genes needed to stop diseases. Further discussion about risk, sir, will be discussed by Ms. Juliet P. Yonko.
Now, this technique presents the following risks. We have unwanted immune system reaction. It means that your body's immune system may see the newly introduced viruses as intruders and attack them. This may cause inflammation and, in severe cases, organ failure. Another one is that targeting the wrong cells. Why? Because viruses can affect more than one type of cells. It's possible that the altered viruses may infect additional cells, not just the targeted cells containing the tainted genes.
What can you expect? Currently, the only way for you to receive gene therapy is to participate in a clinical trial. Clinical trials are research studies that help doctors determine whether a gene therapy approach is safe for people. They also help doctors understand the effects of gene therapy on the body. Your specific procedure will depend on the disease you have and the type of gene therapy being used. For example, in one type of gene therapy, you may have blood drawn or you may need bone marrow removed from your hip bone with a large needle. Then, in a lab, cells from the blood or bone marrow are exposed to a virus or another type of vector that contains the desired genetic material. Once the vector is entered the cells in the lab, those cells are injected back into your body, into a vein, or into a tissue, where your cells take up the vector along with altered genes. Viruses aren't the only vectors that can be used to carry altered genes into your body cells. Other vectors being studied in the clinical trials includes stem cells and liposomes. Stem cells are cells from which all other cells in your body are created. For gene therapy, stem cells can be trained in a lab to become cells that can help fight disease. Liposomes, these fatty particles, have the ability to carry the new therapeutic genes to the target cells and pass the genes into your cell's DNA. The results of gene therapy. The possibilities of gene therapy hold much promise. Clinical trials of gene therapy in people have shown some success in treating certain diseases, such as severe combined immune deficiency, hemophilia, blindness caused by retinitis pigmentosa, and leukemia. But several significant barriers stand in the way of gene therapy becoming a reliable form of treatment, including finding a reliable way to get genetic materials into cells, targeting the correct cells, reducing the risk of side effects, and gene therapy continues to be a very important and active area of research aimed at developing new.
The challenges of gene therapy field. It has evolving for decades despite the best effort of researchers around the world. However, gene therapy has seen only limited success. Why? Gene therapy possesses one of the greatest technical challenges in modern medicine. It is very hard to introduce new genes into cells of the body and keep them working. And there are financial concerns. Can a company profit from developing of gene therapy to treat a rare disorder? If not, who will develop and pay for these life-saving treatments?
Let's look at some of the main challenges in gene therapy. Gene delivery and activation. For some disorders, gene therapy will work only if we can deliver a normal gene to a large number of cells, say several million in a tissue. next main challenges of gene therapy is immune response. Our immune system are very good at fighting of intruders such as bacteria and viruses. Gene delivery vectors must be able to avoid body natural surveillance system. An unwelcome immune response could cause serious illnesses or even death. For instance, the story of Jesse Singer, who had a rare liver disorder that participated in 1999,
disrupting important genes in target cells. A good gene therapy is one that will last. Between 1999 and 2006, researchers tested a gene therapy treatment that would restore the function of crucial gene, gamma C, and cells of the immune system. The treatment appeared very successful, restoring immune function to most of the children who receive it.
One of the challenges of gene therapy is the commercial viability. Many genetic disorders that can be potentially be treated with gene therapy are extremely rare, some affecting just one person out of a 1 million. Just take note of that. Gene therapy could be life-saving for these patients, but the high cost of developing a treatment makes it an appealing prospect for pharmaceutical companies.
Now let's talk about risks to society and direct risks to research participants undergoing stem cell transplantation. Although there may be direct risks to research participants and stem cell transplantation research, there are also social risks. And one major social risk in using embryonic stem cells as a source of neurotransplantation is that new embryonic stem cell lines would have to be created. The derivation of human embryonic stem cells results in the destruction of the embryo, presenting an ethical dilemma for those that ascribe a considerable degree of personhood to embryos.
Other risk of stem cells research is immune rejection of transplanted stem cells. Cyclosporine is a widely prescribed immunosuppressive drug that prevents rejection after transplantation of solid organs or bone marrow, with long-term use shown to cause hepatotoxicity, nephrotoxicity, hypertension, and immune suppression.
The rest of gene therapy is neurosurgical hemorrhage and post-operative infection. It is estimated that stereotactic neurosurgery used in the transplantation of FVM tissue may be associated with a 3% risk of intracerebral hemorrhage and post-operative infection,
Inappropriate stem cell migration and neurological complications. Research participants receiving stem cell transplants may be at risk of experiencing neurological complications owing to stem cell migration from the graft site to inappropriate regions of the brain.
One challenge facing researchers is how to control and regulate the process to successfully trigger differentiation into the desired cell type after the stem cells have been isolated.
Stem cell treatments include new technologies and therapies that aim to replace damaged tissues and cells in order to treat disease or injury by Landval and Kukaya 2006.
The first challenge researchers face when considering stem cell treatment is to understand the mechanisms by which stem cells function in the injured microenvironment using animal models, and then to translate the results of these studies to humans.
The goal of stem cell treatments is to publish all findings and experiences of stem cell treatments, experimentally and clinically, and to open up new possibilities for the treatment of various diseases such as Alzheimer's diseases, traumatic brain injury, heart diseases, diabetes, cancer, and etc., that we may promote health and extend the human lifespan.